Okay, we're gonna pick up here with part three, talking about different types of neurons. We've briefly mentioned and alluded to the fact that there are different branches of the nervous system: the efferent nervous system, the afferent, so the sensory and the motor, and we have neurons that will be a part of that nervous system that either carry information to the nervous system, to the central nervous system, or away from it. Those neurons that carry it to the central nervous system, as we've said, are afferent or sensory. So they're arriving, and these are going to be cells that detect stimuli. So this could be pressure, this could be touch, this could be pain, some type of temperature change. It could be changes in the pH or the acidity level of blood, anything like that that is detecting this information is going to be carried to the central nervous system along an afferent or sensory neuron. So you can imagine when you lean down and touch a hot stove or a hot plate or something that is unpleasant and very hot to the touch, that is detected by sensory receptors that propagate that signal along a axon of a sensory neuron. Okay. Now, if we're staying with our example of touching something hot, maybe leaning on a hot stove, after that happens, it's detected, it travels to the central nervous system, and we get a response. And the response is to pull your hand away, to jerk it away. And the way in which we do this is we have a motor neuron. We have an efferent neuron that is bringing a motor signal down the axon of the motor neuron to the effector, which is our muscle in this instance, to cause it to contract, and it results in some action, right? Some response. So you always have two. In this instance, you have a sensory and then you have a motor. And then interneurons are actually within the central nervous system itself, and they serve to connect the two. So in the example we just gave with the leaning on a hot stove and pulling away, that is a reflex. And there's something called a reflex arc. And so there's a sensory neuron, there's an interneuron or an integrating neuron, and then a motor neuron. And this interneuron serve as a connection between the sensory and the motor neurons, like in an example that we just gave. Now, in terms of the actual structure of neurons, we have a couple of different types. The most common is the one that we've seen before, the multipolar neuron. This neuron is called multipolar because it has multiple poles. Okay, these poles are one axon and several dendrites. Remember, dendrites come into the cell body, and then leaving the cell body, you have the axon. So multipolar is the kind of typical classical neuron you typically see in textbooks and the ones you typically think about. Bipolar neurons, so here we have bi meaning two poles. We have one pole being one axon and the other pole being a single dendrite. We have these in different sensory organs, so things like the eye, ear, and our olfactory organs, so associated with our sense of smell and nose. These are very elongated and have a central cell body, and then off either side you see these two long projections, one being the dendrite that's receiving information, one being the axon that is sending information down to the next neuron or to the target tissue. And then we have unipolar neurons. So multipolar, we had multiple poles. Bipolar, we had two. Unipolar, we're just going to have one. And what you see a single cell body kind of protruding off to the side. And then from this, you get one single kind of branch that runs in either direction. Okay, so this will make more sense when I show you this picture. So here on the left, we can see a typical multipolar neuron, kind of the one you typically think about. We have a cell body with many dendrites coming in. So each of these is a dendrite because each slender process comes into the cell body. And then we have one slender process leaving the cell body as the axon. So again, this is the majority of neurons, like our motor neurons are all multipolar neurons. We have our bipolar, and so here you can see one single dendrite coming in and one single axon leaving, so you have bipolarity, bipolar neuron. Now you may think that all of these are also little dendrites, but the dendrite is the slender process that actually enters into the cell body and connects with it. So you can see in this middle picture with the bipolar neuron, you actually only have one process coming into our cell body, but you have multiple little processes contributing to the formation of that one dendrite. So this is by definition, a bipolar neuron, and then you can see a single axon leaving. So this is like what we find in our retina. And then our unipolar neuron, you see we have a single cell body off to the side, and then you have a little projection, and then two long, slender projections going in either direction away from the cell body. So this is just a single pole. It doesn't have two like we see with the bipolar neuron, and it doesn't have multiple like we see with the multipolar neuron. So unipolar neurons, these are typically sensory neurons that are located, the cell bodies of sensory neurons are located in something called the dorsal root ganglion, and a ganglion is just a collection of cell bodies outside the central nervous system, and we'll take a closer look at that as we progress. So this idea of myelination, if we go back to these pictures here, you can see all of these have this kind of thick black, kind of round bulbous coating over the axon. All of that is myelin. And so this myelination, as we've said in a previous lecture, in a previous part of this lecture, you can get myelination from a couple of different cells, depending on if you're in the central or peripheral nervous system. But the myelin itself is going to be basically the same thing. So in the peripheral nervous system, we have things called Schwann cells, and in the central nervous system, we have oligodendrocytes. The Schwann cells myelinate a single axon by actually wrapping around. The Schwann cell itself wraps around the axon. In certain regions, they leave a space, and this space is called a node of Ranvier. Okay, and this node of Ranvier is the actual point where an action potential is going to take place. It's where it's going to kind of originate, and this is where ions will enter and exit our axon. And everywhere else that is covered by myelin, this will not happen. So what this will end up leading to is the action potential is able to travel very quickly, and we call this quick type of impulse conduction saltatory nerve conduction. Okay, we'll look at a contrasting type called continuous nerve conduction. And then we have oligodendrocytes in the central nervous system, and these may myelinate many axons or several portions of an axon at a single time by extending small little fragments of itself, these little processes, to actually wrap around the axon. So here we can see oligodendrocytes here and here. You see that they're wrapping around, and we know that if we have oligodendrocytes, we have to be in the central nervous system. Here we can see a Schwann cell. It looks slightly different. It doesn't have the process extending out, but the actual cell itself wraps around. And if we see a Schwann cell, we know that we get, or that we are located in the peripheral nervous system. Now, you can see there are regions in between these areas of myelin where there may be a small gap, and it's these gaps that are called the nodes of Ranvier, and this will allow for our action potential along these myelinated axons to travel very, very quickly. So because of the myelin, we get gross anatomical differences and differences in appearance. So when you look at nervous tissue, either from a gross perspective or if you look at it histologically, it will look different depending on whether there is myelin present or not. So we call areas of nervous tissue that have myelin, we call that white matter, and areas that do not, we call that gray matter. Now, white matter is actually white. It appears white because of the myelin. The myelin is a kind of fatty substance, and so it gives it this kind of whitish tinge to it. And we know that if you see myelin, if you see this white matter, you know that you're looking at myelin. You know that you're looking at an area that is full of axons of neurons, because we know that it's the axon that is the actual myelinated portion of the neuron. Now, when we see gray matter, sometimes gray matter looks gray. It's really called gray matter just because it's darker than white matter. Sometimes it can look more kind of a pinkish color in reality. But when you look at regions like the cerebrum, the cerebral cortex, in a gross view, it actually is a darker color, and it's because it is composed of gray matter. This gray matter contains regions of neurons that are not myelinated, and so things like the cell body, the soma, dendrites are not myelinated, and then the very end of the axon terminals and bulbs do not contain myelin, and so they're not going to have that whitish appearance, so we're going to refer to it as gray matter. So if we look here, this region here, this is going to be all white matter along the axon. Everything down here near the axon terminals, dendrites, and cell body is all going to be gray matter. So we can see that here with this image. You see, if we look at a section of the spinal cord, this kind of H, this midline section here, this is all gray matter. Everything around it is white matter. And you can see the same thing here on a frontal section of a brain. You can see an outer cerebral cortex full of gray matter. So you know this contains cell bodies, dendrites, and then all of this more internally located white matter, which we know if it's white matter, it contains myelin. And so this is going to contain axons of neurons. You can see the same thing here in this section of a spinal cord, in this actual specimen, this actual brain specimen. You can see this outer kind of 1/8 of an inch or so of cerebral cortex. It's all gray. And then you can see more internally located white matter, which is because of the presence of myelin. So we're going to stop there with part 3, and we'll pick up with part 4 talking about the action potential.